Hi, can you hear me? Yes. Okay.
Okay, can you hear me? Like you can hear her, but like me. Okay, good, finally. Oh my days. Okay, we should wait for people to join because like there's no one here and there was like 200 reactions on the event message.
Okay.
Yeah, we need someone to do that. Handsome, you can like press start show, so it's better. Yeah, yeah. So like until the session actually starts and people join in, can everyone like say in the chat what topics you find hard or not? Okay, the ping happened. Nice. We should be starting in like 2 minutes. Oh my days, 30 seconds. What do you mean? No? Oh my days. I will say what chapters we're going through when we start. But but it's basically like a revision on everything. Like we had hard questions collected from every single chapter. Oh my days. They deleted the role. This is IGCSE. It's not going to be Excel. But like some things are going to help you in Excel, but like I don't know the syllabus for it. So anyways, should we just start or like wait? There's 60 people here.
Yeah, I think we should start and then you just like come in after me. Or can you hear me?
Okay, finally, I'm back. Okay, so everyone can hear me, right? Okay, good. So basically, I will start with the introduction of the session. So thank you all for joining. Today's session won't be just about reading the slides and going through them as usual. We will try to make it as interactive and productive as possible, such as like the questions we have prepared. Everyone can like write their answers in the chat and we can check them later on to see who got them right and who got them wrong. And lastly, we're going to like be practicing a lot of like of the harder questions that y'all found and like sent on that channel for us. Do you have any questions before we start? The PowerPoints are going to be sent at the end. Yes. It is going to be pinned, so don't worry about that. Any questions or are we free to start? It's going to be around 1.5 hours or 2 hours. We don't know. That's the estimate. Okay, we can start.
Okay.
Okay, go to slide number 2. Yes. Okay. So first of all, the tips that are going to help you a lot with answering the paper. We're going to start with reading the question. How should you read the question in a way that you can ensure that you are not wasting time on it and you're getting all the information needed and is required from you to understand to get the right answer? So point number 1, read the question slowly. Don't rush it. You have time. And underline the keywords that are related to the question to help you eliminate the wrong answers. We're going to talk about eliminations of the rules of how to eliminate the wrong answers later on in the next slide. But like it's basically that in every question there's at least like between 1 or 2 wrong answers that you can just spot from the start of reading the question. But don't worry about that now. Point number 2 is reading the entire question before looking at the answer choices. So basically, for this point, a lot of people just read the first part of the question and they're like, yes, this is easy, this I know, and they go on and answer the question. This can be wrong because you can make a lot of simple and stupid mistakes just because you missed 1 point or 2 points from the question that they said and you didn't read. This will cause you to like lose marks that you can easily get. Then, underlining the key terms. It's not only the key terms, it's also the keywords, as you can see in point 3 and 4. For like calculation questions, you should focus on the units, on the quantities they gave you, on the readings you are given, so you can give the right answer because in the calculation questions, they usually have an answer for every single method you try. So you might have tried a method which gives you a wrong answer, but they have the wrong answer in the choices, and you would think it's right. So be careful for that. And the keywords, they usually have it as bold, like not, except, most, best, something like that. These will help you to determine what you are looking for in your answers. Then cross out the wrong options. We are going to refer to that in the next slide, as I said before. And then for the last 2 points, which is for like the graphs and diagrams, try to understand the units, the axis, the independent and dependent variables, and stuff like that that will help you to answer it correctly. And at the end of the exam, try to read the question again, but try to be more careful with it and more slowly so you can make sure that you haven't skipped any important detail at the end. And this is basically a step that you would go through at the end, so you won't waste time that you can use for another question, for example. Anyways, moving on to slide number 3.
Okay, next is the elimination rule, which is a rule that I often use during multiple choice question. So you know that we have 4 options that we can choose as our answer, right? The question structure usually contains two obviously incorrect options, which I prefer to just cross them out at first, one distractor and one correct answer. So when you do a multiple choice question, I want you to cross out the first obviously wrong options and then underline the key details in the two remaining options that you have left. And then you compare the details that you had underlined and choose the best answer. The advantage of this rule is that it will help you to increase your chance of getting the correct answer from 25% to 15%. Next.
Okay, so this is a point that most people just overthink, just as the slide title. So for paper two, it's not going to be hard to not overthink it, but you might get confused because you answered a question that is similar to it, which would give you an idea on what the answer is, but you are also having doubts on that. So don't overthink the answers. If you have if you are sure of two out of the four choices, then you have a 50% chance that you will get the answer correctly. So that's not worrying. Like you can always guess if you don't know, but you can also refer to other points in the question, other topics you remember in your head in the exam and stuff like that. In paper two, the correct answer is usually supported directly by the information in the question. So usually they give you a graphs, for example, they make it clear on exactly how you need to think about it. And then you should try to avoid adding extra knowledge. Like, for example, a question is asking about something, and you remember that something from a topic you studied, and you remember the details of it. But the question has not mentioned the details. So don't worry about the details because the question will always include the information that you need to know to answer it correctly. Like, don't try to act smarter and to add more details and more knowledge to what is needed for your answer. If an option is not clearly supported by the evidence provided, it is usually not the correct one. This is one of the points that you should basically follow up because, as you know, they usually make it clear on the 50-50% choices. So just pick the most logical one in your head. There's always going to be one that you are leaning more into. And then underlying the keywords, as I said before, is going to help you to avoid this mistake, which is overthinking the answer because you would know exactly what the question is asking you because you understood the question correctly. And lastly, this is something that I always tell people because people overcomplicate simple questions. So sometimes you need to think simpler than you are to get the answer. So in the box below, you can see a personal tip that I wrote myself, which is something I believe in, which is basically to not worry about remembering that each question is worth 1 mark. So you have 40 questions. Every is 1 mark. So if you got one wrong, it's not going to affect you as much. And you always have a 25% chance of getting it right only by guessing without canceling the obviously wrong options like we spoken in slide number 3. So you don't need to worry about what the answer is going to be because you're probably going to get it correctly. Slide number 5.
Okay, next is the time management method. So you know that we only have 45 minutes in your test, right? So I will give you two strategies that you would want to use in your test to maximize the efficiency of your test. So the first one is that I want you to spend 20 minutes or to 45 minutes maximum for your answer in the answer sheet. And you will spend 5 to 10 minutes to transfer your answer to the answer sheet. And the time that you have left, you would want to carefully double checking all of your choices to make sure that it is correct and you would not want to worry about it after the test. The second strategy is that you would want to spend 20 to 35 minutes maximum for your answer. And then the time that you have left, you would want to both double checking and transferring the answer at the same time. So what am I saying is that you are reading the answer again, checking the answer and make sure that it's correct. After that, you transfer your answer to the answer sheets. And always keep the hard question first, do it later.
One point about this that I thought is really helpful is that strat 2 is usually the one that you should always try to follow because you will waste less time with copying your answers on the answer sheet than just trying to focus on the questions. So it's more organized for you, and it depends usually on the time you have left, so you don't need to rush at the end and copy something incorrectly by accident. So that's also another opinion from me. Going to slide number 6. Okay, so this is something that is asked in mostly every single paper, which is the experimental variables. So it helps you that you should know the definition for independent, dependent, and control variables because if you know them, you can basically answer every graph question, every experiment question because most of them are asking you about the effect of something made from another something like that. And an exam trick about this is that if the question asks what should be kept constant, look for the factors that could affect the result. So if you know that the experiment is testing the effect of sunlight, for example, on the growth of a plant, then you know that the sunlight is going to be the changed variable, which is the independent, and you're measuring the growth rate, which is the dependent. Something you should keep control is the water volume. For example, you give the plant or the air or the like room they are in, temperature, stuff like that that can affect the result without even noticing it because it will make your result less reliable. Going to slide number 7. And now we're going to talk about the graph questions in more depth. So if you see a graph question, you should basically follow these steps. So first of all, start with reading the axes because the axis is going to show you what is getting measured and the unit and everything you need to know about the experiment. For example, if it's an experiment question, you should also check the scale to make sure that you didn't use the wrong intervals. For example, for calculating the mean or percentage decrease, increase, etc. You should read the question carefully because you should always know what you are trying to look for and have an image in your head of what you are trying to get from the graph before even like looking at it so that you don't get confused by all the values, all the lines, etc. Try to compare your values to logic. So for example, if it is a question about how many people I don't know. Wait, let me try to think about one. So someone asked me this question, which is about there was a question where a graph is estimating the amount of growth a plant can get in one day. And the answers were like 2 centimeter, 20 millimeter, something like that. And there was the last option, which was like 200 centimeter, which you can logically know that it's incorrect because like what plant can grow 2 meters in one day, for example. So just try to use logic to compare it with the answer you think is right to make sure that you haven't missed some point in reading the question and you need to redo it. Last 3 points are basically looking for the trends, which is like if the graph is increasing, decreasing, having a steady pattern, if there is an anomalous result in the table, for example, something like that. And then eliminate the wrong options, which was spoken about earlier, as I said 3 times already. And lastly is the small details that you should keep an eye on, like values, units, decimal places. These can make a difference in the way you choose your answer and can affect if you get it right or wrong. Moving on.
Lastly is the thing that you should memorize key equation for calculation questions. So this is essential because calculation question appears a lot and regularly in every paper. And this is some of the important equation that you should know so you can cook these up. First is the magnification equation. Magnification, magnification is equal to the image size divided by the actual size. Second is the heart rate calculations. Third is the population calculation. And the last is how to calculate the mean of something. I've also included the conversion table in this slide so you can use it on your questions. And one of my tips is to read and underline the units carefully before calculating, because they will play mind games against you. So please be careful.
And that's basically it for the tips and tricks. If you can go to the last slide.
Thank you very much for listening. So next we will go to the hard question, and so that you can actually use our tips to work out hard questions. So this is some hard question that we had asked you to help us to choose it from your papers. So me and Brofro will go through all of it and explain it to you guys.
And one thing I want to mention before we go through is like thank you for everyone who helps us, like Kizuki, for example, and Huzu, who helped us a lot with formatting this slide. And without them, we couldn't have done this because like we have a lot of exams, a lot of stuff to worry about. So they helped us a lot. So thank you again. Anyways, going to slide number 2. So these are the questions that you people sent on the thread that we made under the schedule message. So we're going to go through them and the details on how to think about the answer. So for this question, is going to ask you about the 3 types of blood vessels, and they are drawn, not to scale. As I told you, they have not or like these keywords as bold, so you can worry about. Not worry, like focus about. So, in which order will the red blood cells flow through these vessels in the human body when traveling from the aorta to the right atrium of the heart? Okay, so everyone can like write their answers in the chat, and we are going to check it in a bit. We're trying to make this as interactive and fun as possible, because I know like listening to us yapping about this all the time is going to be boring. Okay, so the answer is B. You are correct. Go to the next slide, please. So an explanation for this is basically that the arteries need thick lumen because they are carrying the blood from the left ventricle, which is pumped using high blood pressure. So the thick lumen will make sure that the arteries can withstand the pressure of the blood without, for example, bursting in your body. And then 2 is going to be the capillaries, which are thin for one reason, which is to ensure a uh, fast diffusion of nutrients from and out of the capillaries. I mean, that's number 3, and number 2 is the veins, which are going to have valves. And if you know what the valves do, it's basically there to make sure that the blood is not going to go back. And that's because the blood is going to have low blood pressure. So basically, we go from 1, which is from where it's pumped from the heart, to the capillaries for blood exchange, and then 2 to come back to the heart. Any questions regarding this one before we go through to the second question? I feel like this is one of the easier ones. The lumen is basically um, the outline of the vessel, the hollow part, yeah. Any questions before we go to the second question? Okay, we can go now, like move on.
Yeah, so this is the next question.
Yes, okay. So this is an experiment which is set up to investigate the gas exchange in an aquatic plant. And you can see there's a hydrogen carbonate indicator solution where the plant is in, and there's light that is coming to it from the left side. So basically, the question is asking you on the color of the indicator after 3 hours of the start, and it was orange from the start. Yes, they are going to get harder, I promise you. Okay, we can go to the next slide, which is the explanation for this. And yeah, the answer is C, and you should know it from like the table as you can see in the right, how photosynthesis causes the color to turn from like yellow to purple after time. And yeah, there's nothing I can explain in this than that you need to memorize this table. But yeah, moving on. Oh, this is me also. Okay. The apparatus shown was used to investigate aerobic respiration in seeds. The apparatus was placed in a dark room. Dark room means that there is no sunlight for the people who were asking this. But yeah, as you can see, there's a ruler, there is a colored liquid, there's wire, which absorbs the gas produced by the seeds, and so on. And the seeds are soaked in water. So all environmental conditions were kept constant. What will happen to the apparatus? Okay, move on to the explanation. So the answer is C. And there is a few reasons for that. So if you remember, you can also see the method we used to answer the question, which is to underline the keywords like dark room, aerobic respiration, which are going to be factors on how we thought of the answer. So a dark room means that there is no light, as I said. That means that the plant is going to respire and not photosynthesize. So that means that the gas taking in is going to be oxygen because aerobic respiration is taking in oxygen to make glucose and CO2. So CO2 is going to be produced, which is getting absorbed by the wire mesh as they told us in the question. And the direction of movement of the colored liquid is going to come towards the seeds. Why you may ask? It's because of one reason, which is basically that when the seeds are going to absorb the O2, that causes the colored liquid to come closer as there's no way to get the O2 other than that tube that they have connected to the seeds. And that's basically it. So far, so easy. Okay, we can move on, I think.
Next is this question. Can you guys please type your answer in the chat? I think there's maybe like 40, 50 questions in total.
Oh,
Can you
by the
guys,
way, a lot of people have said that your voice is low. I don't agree to them, but like maybe you should try to raise your volume a little more so they can hear you clear.
yeah, I tried. They're like, yeah. Okay, I'm looking at the correct answer. Yay. So this is an explanation part. So basically, glucose is filtered throughout the glomerulus. So in this part, from X to Y, the glucose is transported out of the kidney to flow against the concentration gradient. This is because the glucose concentration in blood is higher than the glucose concentration in the kidney tube. So it is going against the concentration gradient. So we can say that this is an actively transported out of the kidney tubule. So since glucose is already transported out of the tube, there's more glucose in X than E in X than Y. So we will choose B. Do we have any question?
I think we can move on.
Okay.
Yeah, she's got it. Oh my god. Yeah. Anyways, this question. So the cholera bacterium produces toxins that causes chloride ions to be secreted into the small intestine. What is the immediate effect of this on the water potential of blood in the intestinal capillaries and on the water potential of the content of the small intestine? What do you think is the answer of this? Okay, so basically there's a lot of different a lot of different answers. Yeah, I told you the questions are going to get harder. Okay, we can move on to the question or like the explanation, I mean.
All right.
So the answer is B. Why is it B, you may ask? So basically, the cholera bacterium is going to secrete the toxins that are going to irritate the lining of the small intestine. This is going to cause the chloride ions to be secreted into the lumen of the small intestine as a defense mechanism. Like I call it that because this is the response to what happens when the toxins are being produced. So basically, this causes the water potential of the small intestine to get lower, which using osmosis is going to cause the water to move out of the okay, wait. It's going to cause the water to go out of the capillaries into the small intestine, which is going to cause your feces to be uh more watery and like less, I don't know. Anyways, so basically this is the effect of cholera. Do you have any questions on it? I'm getting confused by this. Oh my god. Yeah, you are probably right because like they said immediately. So immediately is going to like be that the blood, the water potential in the blood vessels are going to be higher because like immediately it's going to be lower in the. Oh, you checked the mark scheme. We didn't have the mark scheme when we answered this, so yeah. Apologies for that. So the answer is C. And the reason is like the explanation below is correct, but it's not for the immediate effect. They asked for the immediate effect, which is basically the cause of the toxins, which cause the water potential to be higher in the blood capillaries. So yeah, sorry again for this confusion.
Let me just skip to the next. Do we have any questions left?
Yeah, it's C because they said immediate effect. We probably have missed that point, which is which is, let me say that it was intentional because this will remind you of the tips that we talked about earlier, which is reading the question carefully. And we didn't do that here, which is why we got it wrong. Anyways, let's move on.
Okay, okay. So let me read for you guys this question. Which factor does not help to make the mosquito an effective vector of malaria? So this is an O-level question. This is not included in the IGCSE syllabus.
Yeah, she just said that this isn't a part of the IGCSE syllabus. It's part of the O-level syllabus, which is why we didn't write an explanation to it because this is basically an IGCSE server mostly. So yeah, there is a slight difference in the syllabuses. Yeah, the answer is probably B, but like we are like I'm not sure about this because like it's not basically in the syllabus. Yeah, it's E, 100% E. Anyways, let's move on.
Okay, just skip this question. The answer is A. Okay, okay, just skip this one.
Okay, the answer is A, not B, not C, not anything. It's A.
Okay, okay.
Anyways, don't worry about this because, like, as I said, it's all level and not something you should worry about. But yeah, this question: as a human, the diameter of a aorta as it leaves the heart is 36 millimeter. The thickness of the wall of the aorta at this point is 9 millimeter. What is the thickness of the lumen of aorta at this point? Okay, I'm going to give you like 30 seconds to answer it. There's something that people are going to mistake, I'm pretty sure. Yeah, I can see how some people got it wrong, but yeah. Anyways, let's move on to the description part. So the answer is C, which you know the lumen is going to be on all the sides. So you basically just minus 9, except of, I mean, you subtracted 9 and you should have subtracted 18 because like there's 9 on the right side, 9 on the left side. So when you subtract 36 and the 18 is going to be 20, is going to be 18. Then you are going to convert it by multiplying it with 1000, which is why the answer is C. The people who said the answer is B, it's because they only subtracted 9 and not 18. And this is where most of the students got confused in this question. Yeah, that's basically why some students found it confusing. We are taking the 18 because 36 is the total of the entire aorta. And then there's 9, which is going to be 9 from the right side and 9 from the left side, which keeps the 18 in the middle part, which is what we are looking for, which is the lumen, which is the hollow space where like the blood is going to pass. Yeah, it is very tricky. Yeah, the answer is C. We just said that. Let's move on to the next question. Okay, so This question was asked like 50 times at least. I saw it 50 times from students, and it's basically memorization. There's nothing to do, just memorizing the position of the xylem and the phloem. We are not chemistry helpers, so we can't help you with that. Maybe ask a chemistry helper instead. But yeah, what do you think is the answer for this? Okay, we can move on. So the answer is B, as you said, and the only explanation which I would say is the image I have attached under, which is basically the diagram of where the xylem and phloem are arranged in both the stem and the root, and also the leaf. And you should memorize it because, like, it's always the same and it will never be different. A way that I memorized this is that the xylem is always going to be into the inside and the phloem is always onto the outside. But because it is in the inside, that means in the leaf it's going to be upwards of the phloem. This is a trick that I memorized it in. But yeah, it's basically just memorization. There's nothing much for me to explain. We can move on now.
All right. So this is
Chemistry, you can go.
okay. So this is the question that I found that is kind of not familiar to me because I have not met this question before. So I think that it's maybe a good practice for you guys to try to solve it.
And also apologies for like this session not being as prepared as the paper 4 and paper 6. It's because we both, me and Handsome, have a lot of exams on the same day as like the biology one. So we have been struggling to find time to prepare. So we kinda tried our best to like manage our time between the two.
Okay, so I'm looking at the variation of answer here. So the correct answer for this is D. So what I do to solve this question is that I try to plot off the points in the table to the graph so I can compare which is like the point that is far from the ideal mass the most. You can see both A, B, and C are close to the ideal mass, but like D is quite far and it's in the moderately obese part of it. So I think that D is the correct answer, but we don't have the marks in here. So like it's just our answer. Do we have any question on this one? Okay, I'll explain it again. So my method for this question is that I try to plot the points in answer A into the graph. You can see it here. And I do the same thing with B, C, and D. And I compare the points that I already plot on the graph uh, to see if is there anything that is odd. So you can see in the 4 points here, there's A, B, and C, which is close to the ideal mass section, but D is far from it. You can see like there's a lot of distance between these two, you know? So like these the most we would call like they need to change the most to reach the ideal mass.
If you want, I can try to explain it in another way, which might be easier for you to understand. I don't know if you wanna hear it or if this was enough for you. So like, let me know.
Yeah, this kind of experiment can find.
Okay, they understood it. That's good.
Yeah. So the next question is the question about plant cell. So please send your answer in the chat so I can see it. Okay, so I'm looking at the value. There's a lot of answer here. So, just go to the answer. So the answer for this question is A, because when you look back of the diagram, you can see that there's no chloroplast at all. So like, that chloroplast is the key thing for photosynthesis to happen, which is like a part of the cell that located in the top of the plant, right? So like, there's no chloroplast at all. So it must be located in somewhere that is not dedicated to do photosynthesis or having like giving energy to the cells. So like, it's the root cell, which is some of the key characteristic of the root hair cell is that it does not have chloroplast because it is below the ground, so light cannot reach it. So we cannot do photosynthesis in the root hair, in the root cell.
For the people who said that, for the people who said that the answer is B, cuticle is not a cell, which is why it's incorrect. But yeah, you can move on.
Yeah. Oh my God.
Oh my days.
Okay, how can I fix this?
Okay, wait, give me 30 seconds. Try to like distract them. I will try to fix it.
Okay. Just stare at this diagram, guys, so you know your mistake. There's a lot of people do it wrong, by the way. Why?
Bad news that every single slide after this is literally the same.
Oh my
Until like slide number 30. But yeah, you should uh, refresh your site because I fixed it. So refresh.
I think it's good now, I guess. Oh my God, I'm clicking the wrong button.
Oh my days. Okay. Okay, it's fixed. Thank you for it. So, this question is literally one of the questions that I literally enjoy solving. It's basically saying that there's a chromosome that includes 14, like 4,500 bases. There's 1450 G bases, and they're asking for how many A bases there are. Okay, for the next two questions, you are not going to be able to do, like, you're not going to be able to solve and give us your answer in chat because the answer and the question are both in the same slide. But after the next two questions, we can go back to this interactive thingy. But yeah, let's go to the description part. So the answer is A. So we know that the number of G and C bases are going to be identical since they are complementary. So C is going to have 1450 bases, which is the same as G. So if the total of G and C combined is going to be 2,900, then you're going to divide it by 2, which is because A and T are also identical since they are also complementary. So you're going to subtract the 4,500 and the 2,900 to get the left bases for the total of A and T. If the question was asking you about the total of A and T bases, then the answer is going to be C. But you divide by 2 to get the answer for only A, which is A. Pretty easy, I'd say. Okay, let's move on.
Okay. So I'm gonna read to you guys the question. In a healthy human, in which blood vessel would you find the lowest concentration of urea? So the thing is that renal veins carry out blood away from the kidney. Then you know the function of kidney is to filter urea off, like out of the blood. So when the blood go away from the kidney, it it does not have any urea left because it is already filtered out. So that there's a low there's the lowest concentration of urea in renal vein. Do you guys get it? Okay. I will speak it slower. Okay. So the function of kidney is that it will filter urea out of the blood for excretion process. So the renal vein is that the vein carry blood away from the kidney so that this blood is already filtered. So urea would not be presented in this blood because it has already been filtered out of blood. So it has the lowest concentration of urea. Which one is the highest? I would say that the blood after going after the liver and the blood going before it going into
It's
the
going to be C. The highest is going to be C, renal artery, because it's like before it went to the kidney and got like cleaned up and stuff, and the urea was removed. So yeah, moving on to the next question.
okay. This is also the question with the answer. Um, I'm gonna read it for you guys. What is the reason why antibiotics should be used only when essential? This is because um okay um if we are using antibiotics daily and without following the prescription, like which we just buy it from the hospital and we use it, it will increase the exposure of bacteria that to antibiotics. This will change the selective pressure force mutation in one of the bacteria genes to occur. I would say that the bacteria like adapts to the antibiotics, so they change something in their genes to make them resistant to that antibiotics, which make the antibiotics like harmless to them, and they can spread the genes throughout the population, so that it will increase the resistance of the bacteria to the antibiotics. So this will reduce the natural selection of antibiotic resistant bacteria because the bacteria does not have the resistance yet. It will be killed by the antibiotics and or let, let, left the most antibiotic resistant bacteria in our body. So it will reduce the natural selection of antibiotic resistant bacteria. I think it was so hard. Do you guys understand it?
Yeah, they do.
Thank God.
Okay, so this question is about the osmosis effect. So the diagram shows the water potential of three plant cells. The more negative the value, the higher the solute concentration. What is the immediate movement of water that will occur between the three cells? Okay, so the answer is going to be A. Can you go to the explanation part, please?
Yes.
Okay, so we know that osmosis happens from higher water to lower water potential. So here, the more the negative value is, which is going to be the higher the water potential is, because they said the more negative it is, the more solute it is, which is R, as you can see. If you can go back to the diagram so I can show them. So you can see how R is the least negative. So it's going to go from P, which is the most negative, to Q, is going to go to Q, and it's going to go from R to P and Q, so that both of them are going to be equivalent to R. The effect, can you go back to the explanation, please? Okay, so water is going to move from R to P and Q, while water travels from P to Q as well, because like Q has the lowest water potential, water molecules will travel from the highest to the lowest, so that the water potential between the cells is going to be relatively equal. Do you understand it? I think this is like tricky because they give us negative values instead of positive ones, as you can see. But yeah, let's move on.
Okay.
Okay, so a student investigates the breakdown of fats in milk by lipase. Four test tubes labeled A, B, and C and D are set up. The table shows the content of each test tube. In which test tube will the content be most acidic? The quickest. Yes, it's recorded. What do you think is the right answer? These are the hard questions that people have asked us to do. We had a thread open since like last week, and people have been sending questions there, and we added them all into a spreadsheet. Not spreadsheet, I mean like a presentation. Okay, we can move on to the explanation part. A lot of people have answered it correctly. Okay, I don't know why the slide is red. We haven't done that, but the answer is going to be A. So when fats are broken down, it releases fatty acids and glycerol molecules. A is right because it's the right answer because it contains milk, bile, and lipase, all three. But also, bile is needed to emulsify the fats into smaller globules, and lipase is the enzyme that is used to break down the fatty acids. B, C, and D are all wrong because the presence of bold lipase is going to mean that its active site, which is denatured. So the substrate enzyme complex is not going to be formed, which means that the fats are not going to be broken down. If you didn't get any question, feel free to DM me. I will explain it to you on DMs after the session. But yeah, that's why the answer is A. Any questions before we move on? Hopefully the next slides are not going to be weirdly formatted like this one. I don't know what happened to it. But yeah, hopefully it's going to be better. Yes, you are getting the slides. Okay, let's move on.
Okay. Oh, this is submitted. This is a pedigree diagram question, which is kind of tricky and take a lot of time. So I will give you guys plenty of time for this question, which 90 seconds is that okay? So just solve it and type your answer in the chat box.
We have like 3 or 4 questions like this, so you probably will understand it after it. But yeah, we have detailed explanation for each one of them. So hopefully you can learn it. I think we can move on into the explanation part now.
Okay, so the answer for this question is D. Um, the method that I'm doing here is quite long, so I will not suggesting the audio to do this, but just take a look at the question first. The question is saying that must be caused by a dominant allele. So in this explanation, I would suggest you to choose to write on all of the answer that the question has given to you uh, in recessive allele first. So I test all of these with different conditions. First is that this is the dominant allele in color red and the recessive alleles in color green. So you can see that in answer A, I got both of my results that dominant and recessive is possible. In B, that is also correct, and in C, that is also correct. But in D, when I test recessive alleles, I come up with the result that there's a non-disease offspring came from two homozygous genes. So this is not possible when we test it with recessive alleles condition. So this must be a dominant alleles. So we will choose these. We will choose D, which is the correct answer for this one.
Any questions?
So I have tips for you to do it the faster way, but I have not done it yet, so please check biology chat tomorrow. Um, if I would answer in this session is that to look at the patterns in the diagrams. So you can spot that this is an odd thing to do when there's a recessive alleles for the disease. And then do you see my cursor? So look at these. This is not possible when this is recessive alleles. Yeah, this is an error checking question. You can see this is the recessive condition. So the disease only appears when there's an homozygous recessive genes, like pairs of alleles. But you only have the small a's here. You cannot make a non-disease offspring if that we do not have any dominant alleles here. Do you get it?
So basically, you just need to test it for the recessive and the dominant alleles, and you will know that the recessive is not possible because, like, you can see how in the second generation there is one which is white and both parents are going to be gray, which is not going to be possible as recessive because you need it to be dominant so it can be shown. That's basically what she was saying. That's good. We can move on now. There's another question which is similar to it. I will give you time to solve it. I love how some people already know the answer because they solved it before and they're just saying the answer straight away. You have 30 more seconds before I go to the explanation part. Try to hurry up, please. I'm seeing a lot of B's and there's some C, but I'm pretty sure everyone is going to understand it after we explain it.
Yeah.
It's just going to be trial and error, as we said. Okay, we can go to the explanation part now.
Okay.
Okay, so by trial and error, you can see how they're telling us in the question that the brown fur is going to be dominant and the white is going to be recessive. So that's why for something to be white, you need it to be both small like caps A. We chose A to represent both genes. So recessive, so for one to be recessive, it needs to be double small a. That's how you basically put on every single white one a, which are small. And then you're going to try to use double A capital, and then the one that we used, which is correct, which is why it is the reason for the fur color to change. You can see how um, 2 and 8 are going to be the reason for that because for you can see when 7 and 8 have offsprings, you can see how all of them are going to be white. 8 is going to be AA because, like, if you see the offspring for it, all of them are going to be white and the brown is dominant. So if it was double AA, which is like capital as you are saying, then most of them are not going to be white because it's the dominant gene, which is going to take over of the white one. So like, for example, 11 and 12 are going to be brown, uh, other than like white. So yeah, that's basically how you do it. It's just basically trial and error. You start with listing the recessive, which is going to be double small a. So you put that on every white one. You may say that you can put one capital and one small, but like it's going to be recessive for it to be shown in the phenotype, you need it to be both of them. So that's basically how you solve it. Any questions before we move on? Let me check how many questions. Um, there's 11 questions. Yes, basically that. Or wait, for the female. Okay, no, it's right. Okay, I was about to say something wrong, but thank God I didn't. So yeah,
Okay, should we?
we can move on. Yeah.
Yeah.
Okay. So this question is simple, but it's confusing because the drawing is not really a drawing. It's mostly an X-ray for it. So it's showing a part of the leaf and it's asking you where the xylem is. And as I said before, the xylem is going to be above the phloem. So the answer is B. So yeah, this is an easy question, but it's basically confusing because some people thought it's C, for example, or like A and D, but it's unrelated. So yeah, going to the explanation part, which basically has the same image as the question before. So yeah, this is what you need to know. Just memorize this and you should be fine. So that's it. Moving on to the next one. Okay, this is my part. My bad. So the diagram shows an abnormal sperm cell. Why would the abnormal sperm cell be unable to fertilize the egg? What do you think is the answer for this? For this, also uh, you need to memorize the structure of a sperm cell. So yeah, just memorization part. Okay, we can go to the explanation. So the answer is A. From the opinions, it can be seen that you have to find the missing feature on the given sperm cell structure diagram, which because like they said, it's abnormal. So you're going to compare it to the one that you have memorized, which is the normal one. And you can see that there is an absence of something that is not going to make it possible for you to fertilize the egg. So you can spot out that the nucleus easily by its distinct round shape and the flagellum. You can see them at the end, which is the tail and stuff like that. So what you can't see though is the acrosome, which is basically going to be the membrane, which is located in the front, which is used to dig and go to the egg and like fertilize it. Chromosome is something so different than this. Acrosome is basically a part of the sperm cell, which is basically on the head of it, which is used for like digging, for example. It's a way to explain it. So yeah, chromosome is in the DNA, which is going to be in the nucleus, which is unrelated to this. But yeah, we can move on. We're going to speed run this.
Okay. Next question. Do I need to read this out loud? No. No, you guys just answer it. I'm gonna read your answer. So please type your answer in the chat box so I can see it.
Fun fact that this question got me and Hansen to like struggle on it for like 20 minutes to understand it fully. But yeah, we got it now, so we can just explain it to you without even needing to like waste 20 minutes to just understand it. Well, I haven't studied this in like 2 years. So yeah, I forgot some stuff. But yeah, let Hanson continue with this.
Okay, so I'm looking at a variation of answer here. So our correct answer is C. Also, I'm gonna show you the reason here. You can see that the leaves in the submerged leaves are typically thin. This means that they cut through the water as the current pushes in this way, the direction flow you can see in the diagram, so that they will be thin and they will be stick to the plant. This is also what we call as unstructural adaptation, which allows the submerged leaves to allow water to pass through easily and prevent them from being torn and ripped up. So you can see it's thin, so it's like has a less amount of resistance to the water, so the water can easily go through it.
Okay, to explain why B isn't the answer, a thick cuticle is only shown in the cactus, for example, which is in the desert, which have the function to stop the plants from like drying out because it's going to act as a thick layer to stop the water from evaporating. And here you can see how the submerged leaves are under the water, so they don't even need to have a thick cuticle because like they have infinite supply of water. So that's why B is incorrect. And for D, it's unrelated. So yeah, that's why it's C. But yeah, Handsome have explained it very, 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 very correctly.
Okay, we have that question here. Um, why is A wrong? Um, basically, the fallen leaves cannot. So, why do you guys think that A is wrong? Because the submerged leaves, you can like always see in the beach or like in the lakes that they still have chloroplast in it. The thing is that light can still go through the water. Maybe it's a little, but it still can go through. So the plants still have some chloroplast in the leaves to photosynthesize when they get to the reach of the light.
And because they can photosynthesize, like they have sunlight, they can reach the sunlight, they have water, and there's CO2, there's everything. They have all the conditions that they need to photosynthesize. So that's why it's incorrect.
Okay, do you guys understand this?
We can move on.
Yeah, I think. So this is an explanation part for that question. And this is the next question.
This is your part, by the way.
We don't know. Um, Next question, then, I guess. Okay, so I'm seeing correct answer here with some of the incorrect answer. So our answer is C. So let me explain. Um, After vigorous exercise, which know that it will increase our breathing rate. So, what is the breathing rate? It is the frequency of breaths taken per time, and the depth of breathing increase. This happens because our body needs a lot of air to go into our body for respiration, for the energy. So the air breathed in and out will be significantly greater. This is shown by graph B, C, and D. You can see A is quite tiny, so just cross it out. And furthermore, since the breathing rate increase, we will eliminate B because the frequency of it is just low. So just cross out B. And then um, since this shows a decreased rate, we will construct B and D. Um, since D and C show the same rate as earlier with an increased depth, C is the correct answer because it has the highest frequency with the highest like the amplitude of the wave, so that we can take in most of the air.
Yes, that's basically why. D and C have the same volume, but the thing is, C have higher frequency, which means like more breaths per like 30 seconds. That's why C is higher than D. Because the more you breathe in, like the 30 seconds, the more oxygen you will get in, and like the more volume you will get per 30 seconds. That's basically the concept of it. But yeah, can we move on?
Yeah, this is the explanation of that question.
Okay, so for this one, a student uses two seeds, X and Y, to investigate the phototropism. So there's the diagram. As you can see, there's X, where there's a piece of glass on the left side, and Y, the piece of glass is on the right side. And both of them, you can see that the light is coming from the right, and you can see the before and after sometime. So what do you think is the right answer and why? You don't need to say why, but yeah. I'm going to be the one who's saying why. Okay, let's go to the explanation. Everyone like mostly got it correctly. Let's see, yes. The reason for that is that the piece of glass in X is put on the left side, which is away from the light, and oxygen is produced, which is going to occur on the shaded side, so it will so it can promote the elongation of the shoot to the right side towards the light. That's basically the function of oxygen. But because there's a glass, it's going to be blocking the oxygen to be traveling down that side, which is why the growth is not going to occur. So the shoot is not going to grow towards the light. That's basically why. Any questions? Any concerns? Anything? Okay, we can move on. By the way, just a reminder, try to keep the chat related to the session only. Like don't side chat and stuff because like yeah, it's unrelated to the content we are presenting. But yeah, thank you. Oh, that's me. My bad. I forgot it's my turn. But yeah. So the answer is C. Yes, correctly. You can go to the explanation part. So basically, the vessels are all part of the circulatory system, and an organ system consists of the heart and blood vessels, which are shown in the diagram. It's not going to be an organ like some people said when they asked this question, because you can see there's the kidneys, there's the brain, and there's everything. So it is unrelated to it. So it's an organ system, which is consisted from like groups of organs that have the same function. So yeah. Are we clear to move on? Okay, we can move on.
So this is a question about the circulatory system in the fish, which is a very popular thing that happens in the MCQ question. So please type your answer in the chat box. Okay, I'm looking at a lot of different answers right now. Let's go through the explanation part. Your answer is A. Congrats to someone who gets it right. The explanation for this question is that the oxygenated blood passes from the muscle to the heart, where the heart pumps it to the gills for oxygen, and then the oxygenated blood gets and sent directly to the gills, instead back to the heart. Okay, this is I think this is wrong. I don't know why. So basically, in a fish, the blood will go through the heart first, and then it goes through the gills to get oxygen. When it okay, when it already get the oxygen, it will transfer those oxygen from the gills to the body cells, which is like the muscle. And then when all of the muscle takes the oxygen from the blood, it gets pumped back to the heart. So that's a cycle. I would say that is heart, gill, muscle, heart. So look at all of the answer. We can say that A is the correct answer. Do you guys understand it? Yeah, probably. I am seeing the correct answer here, which is A. So my explanation for this question is that a vaccination consists of the dead antigen or the pathogen's DNA, which is like weakened and it cannot affect our body that much. And when it injected into the body, it will trigger an immune response in our body. So antibody are produced by the lymphocytes, and the lymphocytes divide them into memory cells. This will prepare for the body for an attack by the pathogen in the future, which will happen that in the graph in the question. You can see that A is the correct answer because after vaccination, the immune system produces memory cells for the upcoming attack by the pathogen. So do you guys understand it? Yay. Or not. That's you. I don't know. So this is a question about IVF, which is a topic that already asked in our P4 variant 2. So let's answer this question. This is in the syllabus this year. It is, yeah, and I also see in a lot of correct answer, which is D. In this question, I use a method which is like tick and cross. So you will read off the statement and tick with the one that you feel correct and cross with the one you feel that is incorrect. So my explanation for this is that for the first one, IVF is not available to everyone due to health criteria, age limits, and high cost because the process takes a lot of money. So the first statement is wrong. We cross it out. The second one is that IVF success rates vary significantly based on factors like age and health, and is the sperm or the egg is healthy or not. So we will not guarantee that it is 100% successful. So 2 is also incorrect. We will cross it out. 3. IVF produce an opportunity for single parents to have a family. This is correct because IVF will allow single individuals to use donor gametes to start a family. So statement 3 is correct. Statement 4, IVF is not expensive and everyone can afford it. As I just said that this process costs a lot of money in statement 1. So statement 4 is also correct, incorrect. Statement 5, sperm or egg donation may be involved, which means the couple are not always the biological parents. This is correct because if donor eggs or sperm are used in the process, one or both of the intended parents will not be the biological parent of the offspring. So our answer is D. Do you guys understand this?
Yeah, pretty sure there's no questions. We can move on. Okay. This question had a lot of concerns and misinterpretation for it. If her voice is low, like try to raise it from yourself. I will send you how you can do it. Like just right click on her and then go to the user volume and raise it to the max. And make sure that your volume on your device is also higher. But for me, she's perfectly audible. But yeah, what's the answer for this question? What do you think? I'm going to wait for like 20 more seconds. If you get this question right, you are probably like ready for questions regarding the concentration of this. Okay, I will move on to the explanation part. So basically the answer is B, not C, not D, not A. So if you focused and like paid attention on the question, it said back to the set point, which is back to the normal level. So, which is the level which was before it increased in A. We can see that the level of glycogen is increased above the normal level in A, and the secretion of adrenaline makes the glucose concentration increase, which decreases the concentration of the glycogen to its set point. This means that adrenaline is produced to convert the excess glycogen stored in the liver into glucose, lowering the blood glycogen. I mean, not glucose. There's a mistype in this. Back to normal. This means that A and D are automatically incorrect because they're basically raising the level, and they're saying that it causes it to move towards it, like downwards. So, but C is incorrect because we want to put the answer where the level is lowered to the normal rate, not below it, as it's shown in C. Yeah, I just said that C is incorrect because the concentration below the level. Yes, adrenaline does increase the glucose concentration. We are talking. Okay.
So the next question is this one. Please type your answer in the chat box so I can see it.
If you have a question that you haven't understood, just DM me or like Handsome after the session and we will explain it to you. But we don't have time to go back now and explain it all again. Especially when there's probably going to be a small Q&A at the end. So yeah.
Okay, so I'm looking into the correct answer now. So the correct answer for this question is B. Okay, this is the wall of text. I'm not going to read all of this. I'm just going to simplify for you. So there's 5 stations. I will break it down to station 1, 2, 3, 4, and 5. At station 1, before this sewage outflow, the sewage is clean, and the water is clean too. So oxygen is at its highest, meaning the organic matter in the river is at its lowest. So the sewage outflow is improved after station 1 and before station 2. So in station 2 and 3, like just after um, the sewage gets poured in, so bacteria go into the overdrive, into decomposing that organic material, which why oxygen drops a lot because there's a lot of respiration happens. This means that the organic matter is still high because it still needs a lot of oxygen to decompose it. In station 4 and 5, you can see that it's a start to increase a lot. So that this shows that the river has naturally recovered by the bacteria so that it has keep like breaking down the sewage so that oxygen level rise back up to normal to the set point you can see here. So organic matter is the lowest in here and in 1. Oh, so I think B is the correct one. I just did something wrong.
Do you have any questions before we move on? It's in the pins in that channel. Yeah, you can look at the graph and answer that. But it's better to understand the cause of it because they can bring something similar to this, but without the graph. That's why we are explaining it in details. Like plants in the River. Plants in the river, yeah. We can move on now, I think.
Yeah.
If you scroll down more, you will find the question paper. It's there. I personally pinned it. I know it's there, but yeah, just try to look more for it. If you don't find it, then I'll send it to you. We can move on now. This isn't a specific paper. This has multiple questions, but yeah. What do you think is the answer for this one? This is the question before the last one. Last but not least. Try to answer it. Does no one know the answer? Yeah, probably it takes time. My bad. Okay, I will give you 10 more seconds for the people who are still reading it so that everyone knows. Alrighty, we can go to the explanation part now. So the answer is correctly as B. So um, I'm going to explain why the rest are wrong or something like that. As you can see in the explanation, A is not right because there is no clear trend being followed by both line C, which isn't right because the graph shows a decrease in the cases from 82 in 2003 to 80 in 2004, then an increase in the cases from 80, which is the value you can get from the y-axis, back to up into 81 in 2005, which is why it's incorrect. And D is incorrect because although the number of the cases, because although the number of cases and the vaccination rate both increased from 2005 and 2008, it is still not given that the vaccination doesn't protect the body against it. You can be sure that it's the reason why the you can be sure why it increased also. Even though the vaccination increased, the cases also increased. So maybe there's other variables because as you can see, it's an experiment from 1998 to 2008. You will get the slides in 5 minutes. There is one more question after this. So the answer is B because if you compare the numbers they gave you in the option with the graph, you get that it's correct. Any questions before we move on to the last one? Yeah, I would create a poll, but I don't have permission to do that. The hosting permission role was deleted, so I don't have slow mode bypass or poll or something like that. So yeah, I can do that this time. Maybe next session, maybe next time. But yeah, let's move on to the last question. This is another inheritance question. You already solved it. Oh my God. I'm going to give you 1 minute for this, and then I'm going to explain it. And yeah, that's basically everything for the session. And yeah, we're sorry because some things are not explained as well, but we kinda rushed some parts because of the time limit and exams and all that. So yeah. Okay, we can go to the explanation part now. So the answer is C. Yeah, it's C. And the explanation for that is telling you that the allele for short hair is going to be recessive and the long hair is going to be dominant. So that's a way that you can understand what kind of alleles you're going to perform the trial and error on. So for it to be recessive, which is going to be the gray boxes, best way for this is going to be that every recessive one that is shown, like every gray box or circle, is going to have double small l. Because other than that, if it was one large and one small, then it's going to have long hair because like it's the dominant allele. And when you put the smaller L's on each one of them, you can then try to figure out the alleles for the other ones, which is why it is going to be C because you can see how when you try it, like in the bottom right corner, you can see that there's one which is going to be a circle, which is gray, which means it has the recessive allele. And you can see how above there's two white ones, which have the dominant. So for that to show as recessive for the offspring, you should have it as heterozygous, which is going to give you a 0.25 chance out of 1. We don't know what variant this is because it's different questions in the same slides. So yeah, you can figure out the top bottom right corner because that child is going to be gray, like gray circle, I mean, which is having two small L's. And for it to be possible to have that while they have two parents which have white, is going to be having heterozygous. So that's how you get two out of them. And then for mm, for the second one, like the circle where you see it's going to be white, you can see how the parent for them in the left side, one of them is going to be double L capital, and there is one that is going to be double L small. This is why, this is because like the child is opposite of the other side because the child is white, and then you can see how the parent, the mom, is going to be double small l because she has the recessive allele, which means for you to have your child as white, you need to have the other one as dominant, dominant homozygous, so that your gene is going to be shown on the offspring, which is why that also is going to be heterozygous. For these type of questions, it's better for you Punnett square. I think that's the name for it, where you put the genes and you just do trial and error to figure out which genes are going to work and which ones are not going to work. And that's how you get them. Any questions? As I said, it's a trial and error method. Okay, wait. I think I left by accident. So it's a trial and error method. You're going to try to see if it's going to be homozygous or heterozygous by seeing the percentage of their child being white, for example, or gray. And the one that makes more sense to you is probably the right answer for it. That's how you do it. It's probably like basically, as I said, trial and error and just trying both of them out. You're welcome. So basically, that's all the questions we prepared for you today. That's the end for the slides. We finished all the hard questions. Like if you press enter, you can see that there's no more slides.
There's no more.
Yeah, there's no more. So that's basically it. We're going to have oh my god, you joined and we finished. So we're going to have a quick Q&A. So if you have any questions, you can send them right now and we can try to solve them for you, explain them, maybe some topics. I don't know. We have like 10 more minutes probably. Which one are you talking about? Can you like tell me which one? The double L? Are you meaning the one most to the left? The paper is on Wednesday, not Monday. Yes, you can DM me the question. I will open my DMs and check them. And if I don't check them right now, I'm going to check them later and I will explain them in DMs. I'm pretty sure it's on Wednesday. It's the same day as my A2 math exam. Same day. Pretty sure. I don't know. Okay, different variable. I don't know. I actually don't know. Just check with your like schedule and stuff. Okay, yours is on the 8th and mine is on the 9th. Okay, I got that messed up. My bad. Okay, the most on the left. So it's going to be double L capital because as you can see, the offspring is going to be white. That means that it's going to at least have one large letter L. And you can see how the gray circle is going to be double small L because it's the recessive allele and it's shown to be gray. So that's why it's going to be that. I'm pretty sure you got it now.
I don't think that we have a session for chemistry because we are not chemistry helpers.
Yeah, we're not chemistry helpers.
We're just like biology helpers. So maybe ask like your chemistry helper for the sessions.
Yeah, we don't have the permission to host one.
Yeah.
To explain it again, as I said, the gray circle is going to be double small L. Why, you may ask? It's because it's recessive and it's shown to be gray, which means that it's going to have the recessive allele, which is shown, which is going to be double small L. And for their offspring to have being white, for it to be white, you need the other one to be double L capital. Yeah, you can try with the Punnett square and you will figure it out. Because it's going to be a 50-50 chance. And the other one is not going to be. Okay, I'm going to check my DMs now. Okay, I'm going to forward it to Hansen's DMs because she's the one streaming and she can open it. Okay, just make sure that. Okay, let me delete some of the chats before that. Okay.
No, I just like put it in the slides.
Okay, that's good. That's good. There's someone who said that they didn't get the osmosis question.
Would you
Okay,
say
okay, wait. Someone sent an osmosis question, which is similar to it. It's going to be better.
I saw this one before.
Yeah, okay, this one. Okay, can both of you tell us the mark scheme answers for your questions so we can like tell you how we got the answer? And by the way, the session is over for those who want to leave. These are like just extra questions that you may have and you have asked us to do. Okay, go to the next question. Yeah, extra questions means extra information. You're right. So the diagram shows four mesophyll cells, W, X, Y, and Z, moving through these cells, which arrow shows the direction of movement of water through these cells. What do you think is the answer for this? Yes, the answer is B, because in osmosis, the water is going to travel from like the higher potential gradient to the lower potential gradient. And you see how the least negative, which is negative 350, is going to be the largest number. And it goes down to like the negative 600. That's why it's correct. Do you understand it now? Yeah, that's basically it. No, the least negative, the higher the water potential. Unless in the question they have said otherwise. Like wait, for example, if we added 500 to each of the values, you can see how W is going to be 150, and then X is going to be 100, and you can see how 150 is larger than the 100, so it's going to be from highest to lowest. So yeah, that's the way. Okay, we can go back to the question above it. Slide 62. Yeah, you should explain it, yeah.
Oh wait, what is the title? I don't know this word. Okay. Human gametes, there's a thing that the female gamete is the egg, and then the male gamete is the sperm. So the characteristic of a female gamete is that it cannot move by itself, and it's got a jelly coat on the outside to protect the genes on the inside. And the characteristic of a male gamete is that it has a flagellum to move from the male body to the female body reproductive system, so that it has a flagellum to move, and then it has a mitochondria to like for the energy. It has acrosome as an enzyme to digest the jelly coat of the egg to get inside of the egg, so that we can cross out A because the female gamete does not have an acrosome. We can also cross out B because the female gamete cannot move by itself. And we can also cross out C because flagellum is the thing that the male gamete used to move. So this is like kind of an opposite thing to say. So B is the correct answer because the male gamete has an acrosome and it is motile because it has a flagellum. You guys understand it?
I think that's about it.
Yeah.
We can take like one small question because like we need to end in like 2 minutes. Uh, we're going to send you the slides. You don't even need to worry about that. You're going to get the entire slides. Yes. You know what? I'm going to send it right now before I forget. But yeah.
Yeah, just
Mm, just let me make sure that I give them the viewer access, not the editor one. And yeah, I got it. Okay, I'm going to wait for the slow-mo to end. Just 5 more seconds. And here you have it. You have the entire slide. I'm going to also pin it in the chat. And I'm going to also send the tips one. Okay, I edited the message and added it to it. So yeah, that's basically everything. If you have any more questions, you can just DM us. But yeah. Handsome, do you have anything else to add?
thank you for listening to me and Brofro to in this session. Thank you so much for attending this, and hope you guys can score your maximum score in the paper. That's all.
Yeah, and that's basically how we have hosted a session for paper 4, paper 6, and paper 2. So yeah, thank you all for joining again and have a great evening, afternoon. Anyways, you can leave now. We're going to leave after you guys leave because we're basically the hosts and it's our responsibility that nothing happens until a mode comes and ends the stage. So if you may leave, that would be appreciated.
Okay.
Okay, no one is leaving. Oh my god. The session is over, guys.
Oh my god. Bye. Should I leave?